In this video, we will discuss about the border cell migration in Drosophila melanogaster. So, in normal terms, this is a one kind of fly. And why we are discussing this border cell migration in Drosophila melanogaster? Because it is somewhere related to how the cancer cells migrate, especially in the case of breast cancer cells metastasis that is shown in the panel C. So before going into the lower panels, let us understand the border cell migration in Drosophila. So this is the stage 9 egg chamber, stage 9 egg chamber where the metastasis of these border cells are going on, and this border cells is migrating into this oocyte, and the way these border cells are migrate is with the help of these nerve cells. These nerve cells helps to squeeze these border cells so that they can move from left to right directions in this figure and then go into the oocytes. And these oocytes are been surrounded by epithelial follicle cells. So if we see the zoom in view of this this cluster, we can see that it is a collection of two different cells that release different cytokines, and some of these cytokines are responsible. Some of these cytokines are responsible for the migration of this cluster in these directions, that is from left to right in this figure. And and the reason how and along with this cytokines, they also release this E-caterin. And since at this moment the cells does not have the ECM, there is no ECM has been observed in these nerve cells, so these clusters will not face any resistance from the ECM while moving from one directions to another directions, from left to right directions, in this figure, according to this figure. At the same time, this E-cadherin interacts with other cells and helps in the cluster formations or this mechanical force tractions between the neighboring cells. And because of the cytokines, what will happens, they will lead to signaling pathways activation, and that is being shown here, that this EGFR will get activated through this SPIKN and GRK complex, that will leads to this GTP-RAC complex formations, and that will helps in this affectin polymerization and proteation. So this actin polymerization proteation leads to this, or helps in the migration of this cluster. And how this actin polymerization and proteation helps in the migration, we have discussed that thing in our previous videos in terms of the extracellular matrix. However, in this case there is no extracellular matrix, cells will face less resistance towards migrating from one position to another position. At the same time, this protection will leads to this E-catherine and adhesions and tension. So this E-catherine will also be increased, and because of that, these cells will be able to interact or remain in the form of cluster and then migrates towards the oocyte. And if we compare this phenomena or this process with this cancer cells, we can see that cancer cells also follow the similar process. That is, they also have this two different polar cells that helps that in combination helps in the migration of the cancer cells from one location to another location. That is shown here, is that the border cells are been stained here through this with the help of this FITC. And after that, when they see this the two different polar cells, or when they see these motile cells and non-motile cells, they can clearly differentiate. See, we can clearly see that the breast cancer motile cells is migrating towards these non-motile cells. Non-motile cells are labeled here with the help of with this red color. And in the zoom in view, we can see that in the case of breast cancer, there are two polar cells that can be seen here. So this panel C indicates that the motility of the border cells or the breast cancer metastasis, and the panel D indicates that this two different polar cell formations occurs within the cluster that can be seen here, and more zoom in view can be seen here is that the end portion of this cluster, we can see that there is a non-motile luminal cells, that is this region is non-motile luminal cells, and this region is motile basal cells, or because of the affectins polymerization and other cytokines, other change in the signaling pathways, this region is the motile basal cells, or this region helps in the metastasis of the breast cancer cells. So this comparisons in uh, further made their hypothesis strong that this uh, breast cancers, not just breast cancer cells, cancer cells uh, migration phenomena can be easily understand by by studying the general phenomena of the small, the general phenomena of the other organisms, such as in this case the Drosophila, border cell migration in Drosophila melanogaster is in line with the cancer cells metastasis, for especially to this breast cancer cells metastasis. So I hope you got a basic idea how this border cells migration in Drosophila melanogaster is related to this breast cancer metastasis. If you need to understand more about such comparisons in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.